Welcome back, guys. Today we're going to start this discussion on this topic in biochemistry, gluconeogenesis. Okay, previously we discussed glycogenesis, glycolysis. This channel you can find this video. So let's start with this discussion of gluconeogenesis. Okay. Before this, we are going to see some stages of fasting. Okay, so fasting between 14 to 16 hours. Okay, this is early fasting. Okay, in this early fasting state there is glycogenesis, breakdown of glycogenolysis. This is an hepatic, breakdown of hepatic glycogenolysis. Okay, this is early fasting phase in 14 to 16 hours. Okay, then there is 16 to 48 hours. Okay, this is only fasting phase. That was early fasting, this is only fasting. Here in the 16 to 14 hours phase of fasting takes place our gluconeogenesis. Okay, this gluconeogenesis process is the, remember, donor of ATP. To whom? To, remember, fatty acid oxidation process. Gluconeogenesis is the donor of ATP to fatty acid oxidation process. So we established that gluconeogenesis takes place in fasting state, not early fasting, fasting state. Fasting for 16 to 48 hours. Okay. Proceed with this, the definition. The definition of gluconeogenesis. This is the process in which glucose is synthesized from non-carbohydrate sources. Okay, very important word, non-carbohydrate sources. Synthesis of glucose where in fasting phase of 16 to 48 hours from non-carbohydrate sources. Now what are the non-carbohydrate sources? Okay, non-carbohydrate substrates or sources. Okay, first is the alanine. This is the glucogenic amino acid. Okay, principal glucogenic amino acid. First is the alanine. Okay, next is our lactate produced from pyruvate during anaerobic glycolysis or lactate. Next is, remember, fat. What part of fat? Glycerol, part of fat. Okay, last but not the least important, remember, propionyl CoA. Propionyl coenzyme A, where from odd chain fatty acid oxidation. Propionyl CoA from odd chain fatty acid oxidation. Remember, important note here, acetyl CoA is never used as substrate for gluconeogenesis. Very important fact. Sometimes get confused between acetyl CoA and propionyl CoA. Acetyl CoA is never used. Propionyl CoA, glycerol part of fat, lactate, and as always alanine. Okay. Now this site of gluconeogenesis. Our all important site, liver, and along with it kidneys. So in liver and kidneys gluconeogenesis takes place. As the organelle. Okay, cytoplasm. 3 organelles are involved: cytoplasm, mitochondria. How mitochondria is involved, we'll see, and soft endoplasmic reticulum or smooth endoplasmic reticulum. Smooth endoplasmic reticulum. These 3 organelles are involved in where liver and kidneys. So before going full-fledged into the gluconeogenetic process, first we'll see some cycles. Okay. First is Core's cycle. Okay. Core's cycle. Simple cycles we'll see. Okay. This Core's cycle involves 2 organelles. Okay. First is skeletal muscle, then the liver. Okay. So in skeletal muscle we have glucose. When we exercise, we convert it to pyruvate and further to lactate. Okay. Now this lactate is going to where? Liver. Here this lactate is converted back to pyruvate, converted back to glucose. Then this glucose in a cyclical fashion returns to the skeletal muscle where it is taking place, or skeletal muscle. Skeletal muscle. Lactate formed during exercise is going to where? This is our liver. So 2 organelles are involved: skeletal muscle and liver. Lactate and then liver converted back to pyruvate and to glucose, just then return to muscle. Now another source of this lactate is our all important RBC. Okay, here also lactate is produced anaerobically. You know, RBC is only capable of anaerobic due to lack of mitochondria and other structures. This lactate also enters the liver. Okay, so this cycle is involved in stages of skeletal muscle exercise. Involved in stages of skeletal muscle exercise. Skeletal muscle and liver are involved. This is Core's cycle or Core's lactate cycle, also called Core's lactate cycle. So one cycle is done. Next cycle we are going to see is Cahill's cycle. Cahill's, sorry, I'll write this again. Cahill's cycle. Okay. Here 2 organelles involved are skeletal and muscle. Skeletal muscle and liver. Okay. But the products here are different. So we know in the skeletal muscle we have glucose is converted to pyruvate. Now this pyruvate is converted to, remember we have seen before, the alanine, the principal gluconeogenic amino acid. This process of pyruvate to alanine, this process is, remember, transamination. Okay, transamination we are going to discuss in details in the amino acid metabolism part. Glucose to pyruvate, which is converted to alanine by process of transamination, where this is taking place in skeletal muscle. Okay, now this alanine goes to liver, converted back to obviously pyruvate, which is finally converted back to glucose, and then in a cyclical fashion it goes back to where our skeletal muscle. Skeletal muscle. The importance of alanine will learn in amino acid metabolism. It takes place in the skeletal muscle, and this phase takes place in our liver. Okay, so this is called glucose alanine cycle. Here it was glucose lactate cycle or lactate cycle. Here it was glucose alanine cycle. So these two cycles are very important in understanding how these two, lactate and alanine, alanine is the principal gluconeogenic amino acid and also the lactate, how these two substrates enter into liver where gluconeogenesis takes place. Okay, Cori cycle and Cahill cycle. Cori cycle, lactate cycle; Cahill cycle, glucose alanine cycle. Okay, then number 3, we are going to see the glycerol part of fat, how the glycerol part of fat is involved. So here we have triacylglycerol or TAG. Okay, this is by degradation converted to fatty acids and glycerol. Okay, now this glycerol has to enter into gluconeogenic pathway. How? This is converted to glycerol 3-phosphate. Phosphate is utilized, so an ATP will be utilized here to convert it to ADP. The enzyme will be glycerol kinase. ATP is used, glycerol kinase enzyme, glycerol 3-phosphate. Now this glycerol 3-phosphate will be converted to dihydroxy acetyl phosphate, dihydroxy acetyl phosphate. Now the enzyme is glycerol 3-phosphate dehydrogenase. Remember, glycerol 3-phosphate dehydrogenase, not glyceraldehyde 3-phosphate dehydrogenase. Glycerol 3-phosphate dehydrogenase. This compound will enter into gluconeogenesis. This is how glycerol part of fat enters into gluconeogenic pathway. This is not much important part. Remember, these two cycles are most important to remember: Cahill cycle and Cori cycle. Cori for lactate, Cahill for alanine. And the last part we are remaining with is propionyl CoA. So we have this propionyl coenzyme. This is getting converted to D-methyl malonyl coenzyme. Okay, and the enzyme is propionyl coenzyme carboxylase. Propionyl coenzyme carboxylase. The carboxylase adds the carbon dioxide molecule with the consumption of ATP being converted to ADP with an addition of carbon dioxide molecule. Remember, this phase, this reaction requires biotin. The significance of this will come across in the regulation of gluconeogenesis. This entire process requires biotin. Okay, this is a 4-carbon compound, dimethyl malonyl CoA. This is getting converted with the help of racemase enzyme from a racemer from L-methyl malonyl coenzyme. L form and D form with the help of racemase enzyme. This is also a 4-carbon compound, which is then converted to L, then converted to succinyl coenzyme. Succinyl coenzyme. And here the enzyme is methyl malonyl coenzyme mutase enzyme, and here requires biotin B12 converted to succinyl CoA, requires biotin. Okay, succinyl CoA is further getting converted to oxaloacetic acid. oxaloacetic acid, finally entering to gluconeogenetic pathway. Important fact to remember here is this crucial step, methylmalonyl coenzyme mutase enzyme, requires biotin. Okay, so here deficiency of B12 biotin. Okay, B12 is not exactly biotin, forgive me. B12 is cobalamin. Only B12 is required. Deficiency of B12 will lead to accumulation of methylmalonyl CoA. Methylmalonyl CoA in blood. We have also discussed this whole process in the vitamin B12 B9 discussion. Go check that out, that video. Okay, so this is all about how propionyl CoA converted to oxaloacetic acid and entering into gluconeogenetic pathway. Okay, now I've seen all the 4 important gluconeogenetic substrates entering into gluconeogenetic pathway. Let's start with the discussion of steps of gluconeogenesis. So we will start this by writing the key enzymes of gluconeogenesis. Okay, now this is some relation of gluconeogenesis with the glycolysis, so we'll use that relation further understanding this, how these enzymes are, these names are formulated. We sometimes believe that gluconeogenesis is the complete opposite of glycolysis, but it is not entirely true. We are going to debunk that theory here. The first step, so before the first step, I'm going to demarcate something. Here we will have the irreversible steps of glycolysis. Irreversible steps of glycolysis, and here we have key enzymes of gluconeogenesis. Key enzymes. Okay, first irreversible step here, I'm going to see this formation of pyruvate from phosphoryl pyruvate, to pyruvate, and the enzyme pyruvate kinase. Kinase enzyme, pyruvate kinase enzyme. Here 2 ATPs are produced. Okay, in relation to this, we have from pyruvate formation of oxaloacetate. The first key enzyme here is pyruvate carboxylase. Pyruvate carboxylase, and the name suggests will be addition of carbon dioxide and requires biotin. Again, carboxylase enzyme requires biotin. Our vitamin B7 requires biotin. Here ATP converted to ADP. This is all important step. Remember all the facts are given here. ATP is converted to ATP. It's a 3 carbon compound, addition of carbon dioxide instead of 4 carbon compound. Okay, this is the first regulatory step in mitochondria, one of the organelles where gluconeogenesis is taking place. First regulatory step in mitochondria, pyruvate carboxylase, addition of carbon dioxide to pyruvate to formation of oxaloacetate. Now we have found oxaloacetate. Now a sub part we are going to discuss here, malate aspartate shuttle. Okay, the major process of this gluconeogenesis takes place in cytoplasm. Okay, but this formation of oxaloacetate from pyruvate takes place in mitochondria. So we'll draw this mitochondria. Okay, this is the mitochondria. Mitochondria here, from pyruvate oxaloacetate is being formed. Pyruvate carboxylase. Okay, very clear. Now this oxaloacetate is being converted to aspartate, important amino acid. Okay, the enzyme is, remember, mitochondrial aspartate aminotransferase. It's a process in amino acid metabolism, but remember the enzyme is mitochondrial aspartate aminotransferase. Okay, along with this, oxaloacetate is being converted to malate. Okay, let me draw this in a dotted line, it will be more clear. Oxaloacetate is being converted to malate. So this is malate and aspartate shuttle. Here the NADH is converted to NAD plus. Okay, NADH is converted to NAD plus. Formation of malate takes place enzyme malate dehydrogenase. Okay, now this malate will go out from mitochondria. So there is a shuttle here. This is called the malate shuttle, and malate comes out to where? This is our cytoplasm, where the gluconeogenesis takes place. Also this aspartate comes out. Both this malate and aspartate, with the help of this enzyme, again malate dehydrogenase and AST, get converted back to oxaloacetate. The oxaloacetate that is formed here is moved back to the cytoplasm from mitochondria through formation of aspartate and malate through the enzyme malate dehydrogenase and mitochondrial AST. So this is the total gist of the malate aspartate shuttle. Shuttle is used to remove oxaloacetate from mitochondria to cytoplasm. The all important note to take here is NAD plus is converted to NADH. NADH is being produced here. Okay, here NADH is used, NADH is produced here. Very important step. Okay, so this was malate aspartate shuttle. Oxaloacetate is removed from mitochondria to cytoplasm. Okay, so this is the starting of our gluconeogenetic steps. Okay, but we are not going to write step here. We will write the steps from a little bit further. Now as oxaloacetate has come out to cytoplasm, a 4-carbon compound, it is acted upon by phosphoenol pyruvate carboxykinase. Or all important PEPCK enzyme, phosphoenol pyruvate carboxykinase, to form obviously phosphoenol pyruvate. Oxaloacetate is being converted from 4-carbon to a 3-carbon compound. So obviously carboxykinase compound removes CO2. This is removal of CO2, and here all important is GTP is converted to form GDP. Instead of ATP and ADP, GTP is converted to GDP. Here both decarboxylation and phosphorylation is taking place. The single step, both decarboxylation and phosphorylation is taking place. Okay, so oxaloacetate is converted to PEP. Another important enzyme, PEPCK, PEP's carboxykinase, removal of CO2. Okay, so with these two important breakaway steps here, we will finally be going to our irreversible steps of glycolysis and the key enzymes. Okay, so one of the irreversible steps of glycolysis, formation of fructose 1,6-bisphosphate from fructose 6-phosphate. Enzyme is phosphofructokinase 1 enzyme. Okay, now this will be reversed here. Okay, fructose 1,6-bisphosphate is converted to fructose 6-phosphate, and the enzyme here, remember, fructose 1,6-bisphosphate bisphosphatase enzyme, fructose 1,6-bisphosphatase enzyme. Here water is utilized to form an inorganic phosphate. Okay, it's an regulatory step. It's a regulatory step. One of the regulatory step is formation of fructose 6-phosphate from fructose 1,6-bisphosphate, fructose 1,6-bisphosphatase. Okay. Next, after this, we have our formation of glucose 6-phosphate from glucose, the first step of glycolysis. What was the enzyme? Hexokinase. As this was reversed here, this will also be reversed here. The reverse step taken here in gluconeogenesis is from glucose 6-phosphate to glucose 6-carbon glucose. The enzyme, as this was here, fructose 1,6-bisphosphatase, here is glucose 6-phosphatase. Very simple. Phosphatase, fructose 1,6-phosphatase and glucose 6-phosphatase. Here, as the above takes place, water is used to form an inorganic phosphate. Okay, so these are the 4 irreversible steps. Okay, and the key enzymes in gluconeogenesis. Now we are going to see the full-fledged steps of gluconeogenesis. It's a very simple one. Okay, first, 2 lactate are formed, combines, okay, to form pyruvate. This was the last product of gluconeogenesis. We are going backwards to form pyruvate. From pyruvate, we are not forming 2-phosphoglycerate, we are forming oxaloacetate. What was the enzyme? Pyruvate carboxylase enzyme. Pyruvate carboxylase enzyme requires biatin addition of carbon dioxide. Okay, now this oxaloacetate converted to our all-important PEP, or all-important enzyme PEP carboxykinase. Removal of CO2 here. Here it was addition, here it was CO2. Both requires biatin. Okay. Now, as this PEP is formed, next steps are total reversible steps of glycolysis. From PEP we have 2-phosphoglycerate. 2-phosphoglycerate we have 3-phosphoglycerate. From 3-phosphoglycerate we have 1,3-bisphosphoglycerate. From 1,3-bisphosphoglycerate we have 2 times glyceraldehyde 3-phosphate. From here we have fructose 1,6-bisphosphate. From here we have our fructose 6-phosphate. From here we have our second last product, glucose 6-phosphate, and our final desired product in gluconeogenesis, glucose. So we see that here was all the difference between gluconeogenesis and glycolysis. So now we now see that it is not the entire reverse of glycolysis. Okay, these two important enzymes, formation of oxaloacetate and then formation of PEP. From here onwards, it is the complete reverse of gluconeogenesis, which is glycolysis. Okay, yes. So I'm going to write the enzymes here. The further enzymes we have seen. Okay, from fructose 1,6 to fructose 6 phosphate, fructose 1,6 bisphosphatase. Already the two key enzymes, carboxylase and carboxykinase, we have discussed. And from this glucose 6 phosphate to glucose 6 phosphatase. Okay, the thing we have discussed here, four key enzymes along with the relation to the irreversible parts of glycolysis. Okay, and remember another enzyme here in this conversion. Here also 1,3 bisphosphoglycerate kinase enzyme is used as it was used in glycolysis. Okay, now this was the total overall step of glycolysis. First we have to build up all these, the irreversible steps, the malate aspartate shuttle, the Gale cycle and the Cori cycle, how all the four substrates enters. Okay, this is all the building up for the final gluconeogenetic pathway. Now the energetics. Okay, energetics, pyruvate carboxylase process. Here 2 ATPs are produced. Here PEPCK. Here GTP is utilized. So 1 GTP is equal to 2 ATPs. So 2 ATPs are further produced here. Okay, and finally this kinase enzyme, here also 2 ATPs are produced here. 2 ATPs are produced. I have said 2 ATPs are produced, but in reality 2 ATPs are not produced here. 2 ATPs are actually utilized here because this steps takes place in fasting state. Fasting state utilizes energy. So in finally conclusion we write 6 ATPs are required or generated for gluconeogenesis. It's an energy consuming process. Okay, 6 ATPs are required for gluconeogenesis. Again, for 1,3-BPG kinase 2 ATPs are used. For carboxykinase 2 ATPs are used. Okay, and for the carboxylase enzyme 2 ATPs are used. It takes place in a fasting state. Utilizes energy. Okay, this is all about our gluconeogenetic steps, the irreversible enzymes, the malate aspartate shuttle, the substrates, the Cori cycle, Cahill cycle. Okay, now moving on, we will see the regulation of gluconeogenesis. Regulation of gluconeogenesis. Every product formed from any process in our body is tightly regulated by some mechanisms. Okay, so first is the hormonal regulation. Hormonal regulation. Okay, this takes place in fasting state. Okay, fasting state, there is no food in the body, in the stomach, for insulin to be released. So this takes place in low insulin glucagon ratio. Insulin is low, glucagon is high here. So low insulin glucagon ratio. Here the enzymes are in phosphorylated state. phosphorylated state, so are inactive. Which enzyme? Glycolytic enzymes. Enzymes are in phosphorylated state, so are inactive. The gluconeogenetic enzymes are active. Okay. The logical question is asked. Gluconeogenic enzymes are active in low insulin glucagon ratio in the fasting state. Okay. Next is our allosteric regulation. Allosteric regulation. So remember here, we have discussed before, propionyl CO is one of the substrate for gluconeogenesis, but acetyl CO is not used. Why? Because acetyl coenzyme A is an allosteric activator of pyruvate carboxylase. Okay, acetyl coenzyme is an allosteric activator of carboxylase. Again, fructose 2,6 bisphosphate, not 1,6 bisphosphate, 2,6 bisphosphate is an allosteric inhibitor. Allosteric inhibitor of this enzyme fructose 1,6 bisphosphatase for conversion of fructose 1,6 bisphosphate to fructose 6 bisphosphate. Fructose 6 phosphate. Okay, fructose 1,6 bisphosphatase. Who allosterically inhibits it? Fructose 2,6 bisphosphate. So these are two allosteric regulation of gluconeogenesis. Okay, and to see some applied concepts here. I'm constantly told to remember biotin. Okay, how biotin is important here. Okay. We have raw eggs, and we know from vitamin discussion raw eggs contains avidin. Avidin inhibits biotin. Avidin in raw egg inhibits biotin. Vitamin B7. Okay, so pyruvate 2-oxaloacetate, pyruvate carboxylase, addition of CO2 does not take place. Step fails. Leads to gluconeogenetic failure. Okay, due to raw eggs which contains avidin, pyruvate carboxylase is inhibited, and we also know also inhibits PepsiK, phosphoryl pyruvate carboxykinase. Okay, as it also requires biotin. So both is leading to gluconeogenetic failure or inhibition. Okay, this was one applied concept here. Okay, raw eggs which contain avidin and the gluconeogenetic pathway requiring the avidin here for their process. This is all about gluconeogenesis. Important concept to remember here is takes place in fasting state, that is in low insulin glucagon ratio, not an exactly true irreversible process for glycolysis. Key for irreversible enzymes in relation to glycolysis are discussed here. Malate aspartate shuttle, all important. For removal of oxalo acetate from mitochondria to cytoplasm, where the entire step takes place. Okay. Acetyl CO is not used as substrate for the cycle. Then there is core cycle and Cahill cycle, all important, asked in the exams. Skeletal muscle and liver are used. The lactate cycle and alanine cycle, and the various stages of fasting we discussed early on, out of which in the fasting stage 16 to 48 hours gluconeogenesis takes place. All important, it is the donor of ATP for fatty acid oxidation. When we discuss fatty acid oxidation, we will come across this phase. So keep revising this topic, guys. Thanks for watching. I will catch you in the next one. Good luck.